In the intricate tapestry of life, our existence is intricately woven with the fundamental building blocks of our being, our genes. Within the delicate threads of our DNA lie the blueprints that shape who we are, defining our physical traits, our predispositions, and even our vulnerabilities. Yet, as magnificent as life may be, it is not without its imperfections. The human journey is often marked by the hardships and limitations imposed by genetic disorders and diseases, casting shadows upon the lives of individuals and their loved ones. However, within the realm of scientific progress, a remarkable beacon of hope emerges: gene therapy.
So what exactly is gene therapy? This is a gene. Genes are made up of DNA, which are the blueprints to build the enzyme or proteins that do the words for our body. As far as we know, humans have between 20,000 or 25,000 genes. And most people have 2 copies of each gene, inherited from each of our parents. They are the building blocks of our physical makeup, from the color of our hair, athletic abilities, to our immune system. But sometimes, genes aren't built correctly, and a small adjustment to the DNA within our genes, it can change how our proteins work. It can affect how we breathe, walk, or digest food properly. To better understand how genes change, let's learn about it. So our genes are mutating for all the time as a result of chemical processes in the cell, as well as internal influence like ultraviolet light. We can also inherit mutation from our parents, known when we wind up with this mutation, they can sometimes cause diseases, and that's really where gene therapy comes in. If genes are like the blueprints to our body, gene therapy can fill in something missing drawing. Gene therapy is the introduction, removal, or change in genetic materials, specifically DNA or its molecule cuisine, RNA, into the cell of patient to repair or change a gene to treat the specific disease. The transferred genetic materials then change how proteins or a group of proteins is produced by the cell. It can produce level of certain diseases cause proteins, increase production of diseases fighting proteins, or even produce new or modified proteins within the cell. You may be curious how a gene and the genetic materials it holds is transferred to the cell during the gene therapy. Well, a gene cannot be directly inserted into a cell. Instead of carry, also known as a vector, helps deliver the gene. Typically, viruses are used as vectors because they evolve to be very good at sinking into and infecting cells. But in this case, their motives is to insert the gene or genes into a cell. The infectious part of a virus is removed, allowing it to still enter the cell nucleus without making the patient sick.
Gene therapy encompasses a range of strategies that are used to treat genetic disorders and diseases. First is the gene addition, a strategy used in gene therapy where an additional gene is introduced into the cells of an individual to provide therapeutic benefits. Rather than replacing a faulty gene addition involves delivering and expressing an extra gene to supplement the existing genetic information. Second is the gene correction, a strategy used in gene therapy to fix or repair a specific genetic mutation or defect within a person's DNA. It aims to correct the underlying genetic cause of a disease rather than simply addressing its symptoms. The goal of gene correction is to restore the normal function of the affected gene. This can involve fixing a single DNA base pair, deleting or inserting a small section of DNA, or making more complex modifications, depending on the specific genetic mutations being targeted. Third is the gene silencing, a strategy used to reduce or suppress the expression of a specific gene. It aims to silence or turn down the activity of a target gene that is either overactive or producing harmful proteins. By silencing the expression of the targeted gene, gene silencing aims to mitigate the effects of a genetic disorder or disease. Fourth is the reprogramming. It refers to the process of altering the identity or characteristics of a cell by manipulating their genetic programming. It involves changing the gene expression patterns of cells to induce them to adopt a different cell type or functional state. Reprogramming can be employed to transform one type of cell into another, typically to replace damaged or dysfunctional cells with healthy ones. This can be done by introducing specific genes or factors that can override the existing cellular programming and direct the cells to acquire a new identity or function. Last but not the least is the cell elimination. It refers to the targeted removal or eradication of specific cells from an organism or tissue. This strategy is often employed when there is a need to eliminate diseased or malfunctioning cells that contribute to a particular condition or disorder.
One of the primary applications of gene therapy is to replace missing or defective genes in individuals with genetic disorders. This approach aims to address the underlying cause of disease by introducing functional copies of the faculty genes or providing alternative genes that can compensate for the genetic deficiencies. Second, to deliver genes that accelerate the destruction of cancer cells is a promising approach in cancer treatment. This strategy aims to enhance the body's natural immune response or directly target cancer cells to eliminate them more effectively. Third, to supply genes that cause cancer cells to revert back to normal cells, an exciting area of research with the goals of programming cancer cells to regain their normal functions and behavior. While the field is still in the early stages, these approaches hold promise for developing innovative and target treatment that could potentially transfer cancer therapy. Fourth, to deliver bacterial and viral genes as a form of vaccination is a novel approach that aims to stimulate the immune system response against the specific pathogens. While the concept of using gene therapy for vaccination is promising, it is still in early stages of development.
Fifth, to provide genes that promote or impede the growth of new tissue. It has the potential to revolutionize regenerative medicine and tissue engineering. This approach aims to deliver specific genes that can stimulate or inhibit the growth and development of new tissue, facilitating tissue repair and regeneration. Last but not the least, to deliver genes that stimulate the healing of damaged tissue. It has the potential to enhance the body's natural healing processes and promote tissue repair. By introducing specific genes into the damaged tissue, gene therapy aims to activate cellular mechanisms involved in tissue regeneration. This approach holds great potential for improving the healing of damaged tissues in various conditions, such as chronic wounds, bone fractures, cartilage injuries, and more.
Gene therapy has shown significant progress and promise in treating various diseases and conditions. However, like any medical intervention, safety is a critical concern. The safety of gene therapy depends on several factors, including the specific approach used, the target disease, delivery method, and the careful design and monitoring of clinical trials. Here are some key points regarding the safety of gene therapy. First, advancement in safety. Over the years, there have been significant advancement in the safety of gene therapy. Scientists and researchers have learned from the past experience and have implemented improved strategies to minimize risk and enhance safety. Second, target approach. General therapy aims to target specific cells or tissues in the body, reducing the potential for widespread side effects. Third, risk of immune response. Gene therapy often involves introducing foreign genetic material into the body, which can trigger an immune response. This immune response may result in inflammation, potentially leading to adverse effects. However, advancement in better design and techniques are being developed to minimize immune response and enhance safety. Fourth, adverse events. While gene therapy has shown overall safety, adverse events can still occur. These events can range from mild, temporary side effects to more serious or long-lasting complications. Fifth, rigorous clinical trials. Before gene therapies are approved for widespread use, they undergo intensive testing and preclinical and clinical trials. These trials involve thorough evaluation of safety and efficacy, with close monitoring of the patients for potential adverse effects. Last but not the least, regulatory oversight. Regulatory authorities such as the U.S. Food and Drug Administration or FDA and the European Medicines Agency or EMA closely monitor gene therapy research and clinical trials. They provide guidelines and regulations to ensure patient safety, ethical considerations, and specific rigor. While gene therapy has shown significant potential, it is important to note that it is still an evolving field. Close collaboration between researchers, clinicians, and regulatory agencies is vital to ensure the responsible development and safe applications of gene therapies. It is recommended that individuals considering gene therapy must consult with healthcare professionals who specialize in genetics or gene therapy to understand the specific risks, benefits, and potential side effects associated with their particular condition and treatment approach.
Gene therapy represents a remarkable bridge between the mysteries of life and the power of science. It holds the potential to transform the lives of countless individuals by unlocking the secret encoded within our genes. Through gene therapy, we are harnessing the very essence of life to mend, heal, and restore. With each passing day, scientists and researchers unravel the intricacies of our genetic code, uncovering new possibilities for treating diseases, repairing damaged tissues, and enhancing our well-being. As we continue on this awe-inspiring journey, let us embrace the immense potential of gene therapy with humility, responsibility, and empathy. Together, we can shape a future where gene therapy not only mends broken genes, but also mends broken hearts, inspiring generations to come with its transformative power.